Myself, OP Godara. Today we discuss about seed and seed technology and its introductions. Assistant Professor, Seed Technology, Department, Faculty of Agriculture and Veterinary Science. Firstly, we you know about seed. Seed is a fertilized oil, a fertilized oil होता है with viable embryo. मेनली जो एंजियोस्पर्म्स है अपने जो क्रॉप्स होती है वो मेनली एंजियोस्पर्म के अंदर आती है और इनके अंदर जो सीड है सीड के जो चारों तरफ है वो सीड कोट पाया जाता है और इसी कारण से जो मेनली जितनी भी जो क्रॉप्स है अपनी वो मेनली एंजियोस्पर्म के अंदर आती है। सीड टेक्नोलॉजी क्या होता है कि जो सीड है उसका जो प्रोडक्शन है वो किस तरीके से किया जाता है वो मेनली सीड टेक्नोलॉजी के अंदर आता है। तो जो सीड का मेनली प्रोडक्शन है वो किस तरीके से किया जाता है। सीड का जो प्रोडक्शन है वो मेनली डिफरेंट जो हाइब्रिड टेक्निक्स है उनका यूज किया जाता है और उसी के द्वारा मेनली जो सीड प्रोडक्शन है वो किया जाता है। मेनली जो एग्रीकल्चर प्रोग्रेस है अर्ली डेज फ्रॉम मैन हैज बीन द हिस्ट्री ऑफ सीड्स ऑफ न्यू क्रॉप्स एंड क्रॉप वेराइटीज brought under cultivation, so cultivation के अंडर रहता है। In the early days, it was achieved through the cultivation of indigenous but useful plants taken through introduce इसका introduce करना later through the well known techniques of selection है hybridization है mutation है आपका polyplodization है and plant biotechnology इसका जो यूजेज है वो साइंटिस्ट है उनको यूजेज करते हैं और अवेलेबल मेनी न्यू एंड बेटर वेराइटीज है उनका प्रोडक्शन करते हैं। Farmers, all the scientific research would be to little value unless he gets seeds. Farmers को इन टेक्निक्स के बारे में इतना नॉलेज नहीं होता which are genetically pure से high germination percentage and vigor, high purity, sound health etc. Farmer do not get seed processing these qualities. The yield they obtain may not be as expected. The pace of progress in production therefore will largely depends upon the speeds with which they are able to multiply and market good quality seeds of high yielding varieties. सीड टेक्नोलॉजी मेनली क्या होता है कि फर्स्टली जो साइंटिस्ट कोवन है 1973 उन्होंने आइडेंटिफाई किया सीड टेक्नोलॉजी को द डिसिप्लिन ऑफ स्टडी हैविंग टू डू विद सीड प्रोडक्शंस मेंटेनेंस क्वालिटी एंड प्रिजर्वेशंस। तो सीड है वो मेनली प्रोडक्शन किस तरीके किया जाता है और उसका मेंटेनेंस किस तरीके से करते हैं और उसकी जो क्वालिटी एंड प्रिजर्वेशन है किस तरीके से किया जाता है। आफ्टर दैट फर्स्ट ट्राइजर 1975 मेनली जो साइंटिस्ट है उन्होंने तो डिफरेंट तरीके से इसको डिफाइन किया सीड टेक्नोलॉजी एज द मेथड्स थ्रू विच द जेनेटिक एंड फिजिकल कैरेक्टरिस्टिक ऑफ सीड कुड बी इम्प्रूव। हम किस तरीके से इसको इम्प्रूव कर सकते हैं। इट इन्वॉल्व सच एक्टिविटीज इज लाइक वैरायटी डेवलपमेंट किस तरीके से कर सकते हैं एवोल्यूशन किस तरीके से कर सकते हैं इसका रिलीज है वो किस तरीके से होता है सीड प्रोडक्शन से वो किस तरीके से करते हैं इसका प्रोसेसिंग किस तरीके से होता है एंड स्टोरेज एंड इसका सर्टिफिकेशन किस टाइप से होता है। तो सीड टेक्नोलॉजी इज एसेंशियल एंड interdisciplinary science which encompasses broad range of subjects. In its broadcast sense, seed technology includes the development of superior crops, plant varieties, their evolutions and release, seed production, seed processing and seed storage, seed testing, seed certification, seed quality control, seed marketing and distributions and research on seed physiology, seed productions, seed handling based upon modern botanicals and agriculture science. In narrow sense, seed technology comprises techniques of seed production, seed processing, seed storage, seed testing and certification, seed marketing and distributions, and the related research on these aspects. मेनली ये जो इसका जो शॉर्ट समरी है किस सी टेक्नोलॉजी उसके अंदर कौन कौन सी ये जो प्रोसेस वो इन्वॉल्व होती है मेनली इसका जो कॉन्सेप्ट है the distinctions between seed and grains in vitals behind the seminal importance of to agriculture. A seed, strictly speaking, is an embryo, a living organism embedded in supporting all the foods storage tissue. The seed pertains to materials as like seed, fruit, or vegetative propagated materials meant for saving for planting purpose. The essential functions behind the reproductions. The seed when scientifically produced, such as under seed certification, is distinctly superior in terms of seed quality, namely the improved variety, varietal purity, freedoms for admixtures of breed and other crop species. Seed health, high germinations and vigor, seed treatments and safe moisture contents. A grain, on other hands, include cereals and pulses meant for human consumptions. Difference between seed scientifically produced and grain uses seed. तो mainly जो seed है, it is the result of a well-planned seed program. But in grains, it is the part of commercial produce saved for sowing or planting purpose. Only seed result of sound scientific knowledge, organized efforts, investment or processing, storage and marketing facilities. But grain, no such knowledge or effort is required. The pedigree of the seed is ensured. It can be related to the initial breeder seeds, but in grain, in variety purities unknown. During productions, effort is made to rub out of types, disease plants, objectional weeds, and other crop plants at appropriate stage of crop growth, which ensure satisfy seed purity and health. But in grains, no such effort is made, hence the purity and health status may be inferior. The seed is scientifically processed, treated, and packed and labeled with proper load identity. But in grain, the grain used as seed may be manually cleansed. In some cases, prior to sowing, it may also be treated. This is not labeled. The seed is tested for planting quality, namely germination, purity, admixtures of wheat seeds and other crop seeds, seed health, and seed moisture contents. But in grains, routine seed testing is not done. The seed quality is usually supervised by an energy agency not related with production seed certificates agency. But in grain, there is no quality controls. The seed has to essentially meet the quality standards. The quality is therefore well known. The level certifications tag on the seed containers serve as a quality marks. But in grains, no such standards apply here. The quality is known, descriptive and known nouns. Mainly, goals of seed technology or importance of seed technology. The major goal of seed technology is to increase agriculture productions through the spread of good quality seeds of high yielding varieties. It aims mainly rapid multiplications, timely supply. After that, deteriorations causes of varieties and their controls. Firstly, varieties a group of plants having clear distinguished characters which when reproduce either sexually or asexually retains these characters. The main aim of seed production is to produce genetically pure and good quality seeds. But why and how the genetic purity of a variety is lost or deteriorated during the seed multiplications? The several factors that are responsible for loss of genetic purity during seed productions as listed, mainly developments, variations, mechanical mixture, mutations, natural crossings, genetic drift, minor genetic variations, selective influence of disease, techniques of the breeder, breakdowns of male sterility, improper या defective seed certification systems. तो इस प्रकार मैंने जो सीड प्रोसेसिंग टेक्निक्स है वो मेनली डिफरेंट से तरीके से अगर उसका हैंडल किया जाए तभी अपन जो सीड है उसका प्योरिटी रूप में कंट्रोल कर सकते हैं mainly जो शीट की क्वालिटी है mainly Thomson in the 1979 defined sheet quality as a multiple concept comprising several components and their relative importance in different circumstances and laid much emphasis on analytical purity or physical purity, species purity and genetic purity, freedoms from weeds, germination percentage, seed vigor and health, seed moisture contents, seed size, weight, and specific gravity, seed quality characters. A good seed should have the following quality characters: improved variety, genetic purity, physical purity, seed germinations and vigor, freedom from weeds and other crop seeds, seed health, seed moisture, seed size, weight, etc. Seed certifications should have know the procedure. Seed certificate. Of mainly seed certifications. The seed technology. सीड टेक्नोलॉजी है उसका जो रोल है वो फर्टिलाइजर आउटलाइन सर्वर area of the technology, the introduction of quality seeds of the variety wisely combines with other inputs. Yeah, the cultivations of high yielding varieties. They have helped to increase over a period of 40 seed basis to for secure food supply. Successful implementations of the varieties programs in India has led to a remarkable increase in production have been brought down in spite of rapid increase. Principles means to secure crop yields in less favorable areas of production, the supply of good quality seeds of good varieties suitable to these areas is one of the important contributions to secure crops. A medium for rapid rehabilitations of agriculture in case of natural disaster, in case of floods and drought affected areas. The government will provides the proceeds from national seed stocks or rehabilitates the agricultural productions of whole grains in the country. Seeds of new varieties must be made available still in time so that the planning schedule they are able to seeds of high quality of seeds. This is necessary. Seed improved varieties. High quality with various farmers. Natural person is an important source of cultivation and sexually propagated crops and lessons of seeds from delicate structure. Sense of cultivation depends upon the amount of times. Other hand, natural crossing is main source of contamination in those fertilizers or extent of genetic contamination breeding system. Isolation distance, varietal mass, and pollinated agents. To overcome the problems of natural crossing, isolation distance has to be maintained. Increase in isolation distance decrease the extent of contaminations. The extent of contaminations depend on the directions of the wind flow, number of insects present, and their activity. Uh, Minor genetic variations, it is not much importance. However, some minor genetic changes may occur during production cycles due to difference in environment. Due to these changes, the yield may be affected. To avoid such minor genetic variations, periodic testing of the varieties must be done from breeder seed and nucleus seed in self-pollinated crops. Minor genetic variations is a common feature in often cross-pollinated crops. Selective influence of disease, proper plant production measure must be taken against to measure pest and disease. Otherwise, the plant as well as the seed get infected. In case of foliar disease, the size of the seed gets affected due to poor supply of carbohydrates from infected photosynthetic tissue. In case of seed and soil-borne disease like downy mildew and ergot of jawar, smut of bajra, and bunt of wheat, it is dangerous to use seed for commercial purpose once the crop gets infected. New crop varieties may often become susceptible to new races of disease or out of seed production programs, is like Sureka and Falguna becomes susceptible to Galva's by types 3. Techniques of the breeders instability may occur in variety due to genetic irregularities. If it is not properly assessed at the time of release, premature release of a variety which has been bred of a particular disease lead to the productions of resistant and susceptible plants, which may be an important cause of deteriorations. When Sonalika and Kalyan Sona wheat varieties were released in India for commercial cultivations. The genetic variability in both varieties was still in growing stage, and several secondary selections were made by the breeders. Generally in hybrid seed production, so if there is any breakdowns of malleability in many lead to a mixes of F1 hybrids and sulfurs, improper seed certification. It is not factors that deteriorates the crop varieties, but is any other above factors, it has not been checked. It may lead to deteriorations of crop varieties. Field instructions and approval of the crop and at a critical stage for verification of genetic purity, deteriorations of excess weight and seed-borne disease, sampling and sealing of clean slots, growing of samples with the authentic stocks or grow-out test, various steps suggested by scientists in, mainly Hartmann and Kester 1968 for maintaining genetic purity are as follows: providing isolation to prevent cross fertilizations or material mixes, roughing of seed fields prior to planting. Periodic testing of varieties for genetic purity. Grow is adapted areas only to avoid genetic shift in the variety. Safeguard for maintain genetic purity, control seed sort, preceding crop requirements, isolations, roughing of seed fields, seed certification, grow out test. The seed use should be appropriate class from the approved source for raising the seed crops. There are four classes of seed from breeder seed with are given and defined by associations of official seed certification agency AOSCA. Nucleus seeds, it is a handful of seed maintained by consent breeder for further multiplication. The nucleus seed will have all character that the breeder has placed it and highest genetic purity. The quantity of nucleus seed is in kilograms. Breeder seed, it is produced by the concerned breeder or sponsoring institutes or which use for producing foundation seeds. It is 100% genetic purity. The label or tag issued for breeders is golden yellow in color. The quality of breeder seed is assured by the monitoring team constituted by the governments. Foundation seed produced from breeder seeds and maintained with specific genetic identity and purity. It is produced on government farms or by private seed produce. The quality of foundation seed is certified by certification agency. It has genetic purity of about 98%. So the certificate tag or label issue for foundation seeds white in color. Preceding crop requirement, this has been fixed to avoid contaminations through volunteer plants and also the soil-borne disease. Isolation is required to avoid natural crossings with other undesirable types of type plants in the fields and mechanical mixers at the time of sowings, threshing, processing, and contaminations to the seed-borne disease from nearby fields. Protections from these sources of contamination is necessary for maintaining genetic purity and good quality of seeds. The extent of off-type plants in another source of genetic contaminations. Off-type plants differing in their characteristics from that of the seed crops are called as off-types. Removal of off-types is referred to as roughing, and the main source of off-types are segregations of plants from certain characters or mutations. Voluntary plants from previous crops accidentally planted seeds of other variety. Disease plants of type plants should be roughed out from the seed plots before the said pollen and pollination occur. To accomplish this regulation supervision, certain personnel is required. Grow out dust varieties that are grown for seed production should be periodically tested for genetic purity by conducting GOT or to make sure that they be being maintained in true forms. GOT test, grow out test is compulsory for hybrids produced by manually emasculations and pollinations and for testing the purity of parent lines used in hybrid seed productions. Seed quality objective multiplications of quality seeds under vigilance supervisions of breeder of seed certification agency to distribute quality seeds of notified varieties for sowing purpose. Seed is seed of notified varieties are multiplied in for their systems by the involvement of ICR Indian Council of Agriculture Research, state agriculture institutes, national seed corporations, and seed certification agencies. Okay, students.